Constituent factors observed in viral diseases, Part 4 of 5. Pause text and illustrations for detailed study. Refer to the previously posted constituent factors observed in viral diseases, Parts 1 through 3. Protective eyewear can greatly mitigate the more remote possibility of infection via the ocular route. The half-life of persistent ambient aerosolized virus is measured on the scale of hours. Aerosolized virus is capable of traveling at least 13 feet, exceeding common social distancing recommendations. Facial masks can provide effective protection as barriers, lowering the chance of viral inhalation and expulsion by users. Unprotected individuals are at a greater risk of inhaling infectious viral particles or disseminating same if they are contagious. One-use N95 facial filtering surgical respirators are the most efficient and protective mask against aerosolized virus inhalation and infection. Simple paper masks are also protective. Contaminated surfaces and objects are also a source of COVID-19 transmission and infection. Physical contact with these contaminated fomites and subsequent contact with the mouth, nose, and/or eyes may result in human infection. COVID-19 may persist on paper, cardboard, wood, and cloth, plastic and stainless steel, as well as money and glass, for up to 3 and 24 hours, as well as 2, 7, and 4 days respectively. Surgical gloves and garments are also barriers against viral contamination and infection. Hand decontamination using a disinfectant containing at least 60 to 90% alcohol, or washing contaminated or soiled hands with soap and water for a minimum of 20 seconds is preventive of viral dissemination and infection. Dependent on the ambient viral load and circumstances, as well as the degree of best availability and recommended use and practices, these protective measures offer varied levels of protection without an absolute guarantee against infection. Asymptomatic individuals infected with COVID-19 are capable of infecting other people. Viruses exhibit a specific affinity for infecting certain tissues. Such viral tropism is determined by cell susceptibility, host defenses, temperature, pH, and physical barriers. Intact skin is a physical barrier prohibiting the entry of viruses into the body. Body immune defenses are capable of attacking viral infection. Viruses are capable of spreading to diverse parts of the body with time, including the nervous system. The entry of viruses into the body occur at mucosal surfaces via inhalation or direct contact with a contaminated entity. The site of skin punctures with entry into the bloodstream and via ingestion, causing infection in a human or animal host, resulting in new viral production, spread, and the infection of other hosts. A vector is an organism, such as a biting insect or a parasitic blood-sucking mite, that spreads infection by conveying a pathogen from one host to another without causing infection in and of itself. A vector can convey an infectious foreign viral DNA particle to a host cell. In the case of most mosquito-borne viruses, there are no currently known vaccines or medication. Japanese encephalitis and yellow fever are 2 currently known exceptions. Animal, human, bacteria, and plant viruses exist in nature. Antibiotics are sufficiently non-toxic chemotherapeutic agents or medications capable of inhibiting the growth or the killing of infectious disease-causing microorganisms in the host. There is a common misconception that antibiotics are effective against all infectious agents. Antibiotics cannot kill viruses since they specifically and exclusively target the growth and proliferation mechanisms of bacteria. Antiviral chemotherapeutic agents may be prescribed early in the infection process to lessen the degree and duration of infection, since they interfere with the functional biochemical machinery of the virus, affecting the extent of viral infection and proliferation in the host, being potentially curative. Viral infections may result in complications involving secondary bacterial infections, against which specific antibiotic chemotherapeutic agents interfere with the infection and proliferation process of the bacteria, also being potentially curative. Bacteriostatic and bactericidal antibiotics, respectively, prevent bacterial growth or kill bacteria. The result of viral and bacterial mutation are microorganisms that have adapted genetically, resulting in the capability to survive and proliferate despite novel specific environmental and antimicrobial challenges to their survival. Antiviral and antibacterial agents specific to these previous types of microorganisms may no longer be effective against newly mutated forms. This results in what is referred to as antibiotic or drug resistance, whereby a specific infectious virus or bacteria becomes capable of withstanding the effects of a drug capable of killing most members of its species. Immunoglobulins are proteins in the serum and cells of the immune system, capable of participating in an immune response. Antigens are foreign or toxic substances capable of inducing a specific immune system response. Exogenous antigens are derived from the environment, such as bacteria, viruses, as well as irritating pollens and chemicals. Endogenous antigens are produced by the body in a specific response to a viral infection. In its attempt to combat the antigenic challenge, the body's biochemical response is to produce substances that may be curative. Antibodies are immunoglobulins produced in response to viral antigens capable of inducing an immune response at specifically corresponding host cell receptors. Antibodies may be generated against any viral protein with genetic specificity in an infected cell, against the glycoproteins expressed on the virion envelope or the cell membrane, and are of significance in controlling infection. Circulating antibodies that neutralize antigens are key to prevent reinfection in antibody-mediated immune responses. Some antiviral drugs are capable of binding to a specific receptor inhibiting viral attachment to the host cell, preventing infection, viral replication, and exit of new viral material capable of infecting other host cells. White blood cells, also referred to as corpuscles or white cells, are immunocompetent cells. Immunocompetence, also referred to as immunoresponsiveness or immunologic competence, is the ability to develop an immune response following exposure to an antigen. Leukocytes are white blood cells including lymphocytes and monocytes. Lymphocytes are immunocompetent white blood cells comprising the bodily or humoral blood-bound antibody response to pathologic antigens. A B cell is an antibody, AB, producing lymphocyte responsible for humoral immunity. Plasma cells are fully differentiated B cells producing one type of antibody. An affinity exists on the part of memory B cells for a specific antigen, AG. A B cell produces antibodies for specific antigens that it binds via a B cell receptor, BCR. B cells are produced in the bone marrow as part of the adaptive immune system. In the following clonal selection of B cells illustration, an antibody is a Y-shaped protein principally produced by plasma cells, capable of neutralizing pathogenic antigens such as viruses and bacteria. Clonal selection of B cells illustration. Antigen selectively generates activation, division, and differentiation solely in cells expressing receptors with which it is capable of combination in the process of lymphocyte activation. This results from a second exposure to antigen. This pre-existing clonal pool of cells eliminates the antigen. This immunological memory functions in natural immunity as well as the artificial immunity such as that derived from vaccination. An infecting virus and or virus-infected cells can stimulate B-type lymphocytes, also referred to as B cells, that are viral antigen specific to produce corresponding antibodies. Carbohydrates are essentially sugars capable of fueling biological processes in living organisms. Glycoproteins are bonded protein carbohydrate conjugates. Immunoglobulins are glycoproteins functioning as antibodies. A therapeutic immunoglobulin is a sterilized solution of antibodies derived from donor blood of an individual previously exposed to a particular infectious organism who has developed neutralizing antibodies that can engage and defend against this infectious antigen within a time frame after administration to an infected patient. Such convalescent plasma transfusions of antiviral antibodies are associated with increased rates of recovery. Serologic antibody testing is confirmative of patient exposure to a corresponding infectious antigen. Antibody sequences are short-lived and have a characteristic half-life in serum. Certain immunoglobulins and vaccines may be administered after viral exposure to prevent infection development or reduce the severity of infection. Immunoglobulins are also therapeutic in a patient whose immune system does not adequately respond to an infection. Immunoglobulin types A, G, and M are B cell surface immunoglobulins on the host cell antigen receptors that will specifically bind with the particular invading viral antigen, exerting antiviral activity in the humoral immune response. Humoral immunity is mediated by antibodies. Serum consists of the clear protein-rich fluid separating out upon blood coagulation. It is also referred to as the clear portion of any body fluid. Plasma is the fluid portion of blood, lymph, or milk containing various suspended particulates. The interstitium is a minute fluid-filled space between cells. The interstitial fluid in this space is a water solvent comprised of amino acids, cell waste products, coenzymes, fatty acids, hormones, neurotransmitters, salts, sugars, white blood cells, and waste products. It is derived from substances leaking out of blood capillaries, functioning in oxygen and nutrient delivery to, and waste product elimination from cells. Lymph is derived from tissue fluids and contains lymphocytes from all parts of the body. The formation of new interstitial fluid replaces older fluid draining to lymph vessels throughout the body, becoming lymph or tissue fluid that is returned to the blood via this lymphatic system having a circulatory and immune system function. Immunoglobulin A and G are prominent in the antigen-antibody response in serum. Immunoglobulin M is the first antibody to respond upon initial antigenic exposure and is involved in B cell activation. Lysis refers to the dissolution, rupture, and death of an antigenic particle or a cell by a specific agent. Phagocytosis is the ingestion of an antigenic particle into an invagination of the cell membrane, which is encapsulated within this lysosome and taken into the cell, where it is killed and digested by lytic lysosomal enzymes. Ubiquitins are amino acids that attach to proteins, functioning in protein activation, localization, interaction, modulation, inactivation, and degradation affecting cellular processes. When covalently bonded to other cellular proteins, the proteinaceous ubiquitin marks these proteins for proteolytic degradation, particularly by a proteasome in a protein complex process catalyzed by proteases. Immunoglobulin M binds to antigens, marking them for phagocytosis. It is mostly found in serum and the interstitium in very low concentration. Antibody can neutralize invading viruses by blocking viral host cell antigen binding. Cytotoxicity refers to the destructive effect of a substance on a cell. Complement is a blood serum bound protein functioning in cellular dissolution, also referred to as cell lysis, occurring in the neutralizing immune response to viral antigens. Antibody is capable of specifically neutralizing viruses via viral antigen recognition on infected host cells, capable of resulting in antibody-dependent cytotoxic cell or complement-mediated cell lysis. Illustration of cytokine release. Cell-mediated immunity involves the recognition and/or killing of virus or virus-infected cells by leukocytes. Cytokines are non-antibody proteins released by T-type lymphocytes contacting a specific antigen, acting as intercellular mediators of an immune response. Cytokines participate in the direct activation of antimicrobial effector functions, providing regulatory signals that can specify, amplify, and resolve immune system responses to infection. Cell-mediated immunity also involves the production of various soluble cytokines by leukocytes when triggered by virus or virus-infected cells. Cluster of differentiation CD markers are cell surface molecules of leukocytes and platelets that identify the particular type of leukocyte. There are four major kinds of T cells. CD4 plus type glycoprotein T helper cells function in B cell maturation into plasma and memory B cells. CD8 plus type glycoprotein T cytotoxic cells function in the lysis of virus-infected and tumor cells. Antigen-specific memory T cells persist after an infection is eliminated by the body, rapidly converting into effector T cells upon subsequent exposure to a specific antigen, providing an immune response. Natural killer cells are cytotoxic T cells that kill virus and tumor cells. NK cells are part of the innate immune system provided for by the genetic composition of the host. Bodily immune cells and chemicals and physical barriers, such as the skin, that protect against and respond to foreign elements invading the body, are examples of innate immunity. Active immunity consists of the acquired immunity developed by the body and its capability to respond to specific antigenic challenges. Illustration of the primary immune response illustration of clonal selection and expansion of T lymphocytes. The major histocompatibility complex, MHC, is a genetic region in all mammals whereby large proteins in immune system cells are capable of identifying compatible or foreign proteins. It functions in cell signaling between lymphocytes and cells expressing antigen. MHC signaling between lymphocytes and antigens is the most effective between these elements sharing the given complex. MHC also plays a role in the matching of suitable organ or bone marrow donors with recipients, as well as rejection of incompatible grafts between such parties. Major histocompatibility complexes 1 and 2, MHC1 and MHC2, are proteins that present peptides on the cell surface for T cell recognition. MHC1 glycoproteins present endogenous antigens from the host cell cytoplasm to cytotoxic TC cells. TC cells are T lymphocytes that kill infected, damaged, and cancerous cells. MHC2 glycoproteins are antigen-presenting cells that present exogenous antigens from foreign sources such as bacteria to CD4 positive T helper cells for the initiation of the antigen-specific immune response. CD4 positive refers to the presence of this protein in this type of T cell. T cells are produced by stem cells in the bone marrow and mature in the thymus, which is a lymphoid organ in the chest. T lymphocytes are part of the adaptive immune system. Cytotoxic T cells and natural killer cells are lymphocytes capable of recognizing and destroying virally infected cells. Some tumor cells resulting from viral infection can also be recognized and destroyed by this mechanism. Natural immunity is mediated by cells capable of immunological activity without immunization or antigen specificity, such as natural killer cell activity against viral infection and tumor cells. Phagocytes are cells capable of ingesting and destroying particulate matter, including antigens mediated by specific cell surface receptors also involving complement. Macrophages are specific phagocytes also involved in the antigen-antibody response that recognize and destroy virus-infected cells as well. Monocytes are phagocytic leukocytes transported to tissues where they develop into immunocompetent specific macrophages. Lymphokines are soluble cytokines, not comprising an antibody or complement, released by antigenically sensitized lymphocytes that mediate immune responses. Cytokines that are produced by monocytes are referred to as monokines, as well as natural killer cells functioning as lymphokines. Both function in the development and regulation of immune responses. Viral antigen antibody, as well as related T cytotoxic and helper cell activity, can involve humoral and cell-mediated hypersensitive immune system reactions. Proceed to part 5 of 5.